So what did I want to say? So today we have a guest lecturer. His name is Dilan Jaunky. He's been a long-time friend since, let's say, 2002, I guess. We did elementary school together, high school, karate, university, master's, PhD, and now we're here. Pretty much. So he took the business route. He was in high school. He was one of the top students. I think he was a top student. I was busy with this lady that was taking care of troublemaker students. I think she had like 20% of her time was just me, you know, always in detention and suspended. This guy though was killing it in martial arts, doing international competitions, studying, acing every exam, the whole thing. And he proved pretty much everyone wrong on the fact that if you are studying very hard, you can't build a business because you're not a B student or C student or whatsoever, okay? You probably saw that on TikTok, right? So study hard, build a business, okay? And then we came here to Concordia. We studied together. We did our undergrad together, 261. That's where we started studying like all the time together. I was gonna show a picture of him sleeping in class 225, but I was like, you know what, let's keep it professional, you know? And then we had, actually I remember, this is a funny story. So in Biochem 1, it was the 14th of February 2013. So yesterday, we were doing a lab, okay? And it was at 7 p.m., okay? From 7 to 10, I think. And I was super tired. So I had the cuvettes all placed like one next to the other. And then I hit them. I destroyed the whole experiment, okay? He got so mad, he made me wash the dishes, the whole lab, okay? In any case. Yeah. But anyway, so he did his PhD here at Concordia with Lipid Tech Inc on cancer. We worked in the same lab. Then he went to Charles River. He was a study director there. Started his business. Now there are 4 people in the business and trying to get big investments at the moment. They built a technology that he will talk to you about, and hopefully you like it. And at the end, if you listen, there's an assignment for you. Cool? So let's be nice and welcome our little buddy here, Dilan. There you go.
Can you hear me now?
Well, let's put it a little higher.
So I might cough a few times. I have a sore throat. My newborn was sick, so I got his whatever he has. So that's a really, really great intro. I guess now you know a lot more about me than I expected you to know, beyond the scope of just this guest lecture. So yeah, it's true. We spent a lot of time, pretty much, our academic routes were pretty much in parallel for most of the time. We left Concordia and then we're right back here doing very different things, but still under the same kind of institution. Because that institution, which is Concordia University, is always growing and expanding its reach to students and to now entrepreneurship. So this is why I came back, because entrepreneurship became a big facet of what I wanted to do with my career. I explored academia, understood academia, enjoyed it, figured out that that was the biggest kind of plus I could have in terms of a research component, but then I needed to see the other side of the coin. What do you do with all of this knowledge and how do you make it relevant to the general public and pretty much our world market? So this is the intention of this lecture is really to be, a little bit open-minded about the future of what you're technically learning right now is step 1. Where can all this knowledge put you later on, let's say 10 years from now? And the title of the lecture is Coded to Coding, is to show you genetics has evolved significantly from the time, when we started learning about the fundamentals of what the ATCG is, nucleotides, the structure of DNA, to what we're doing right now today in the real market. So if ever you have any questions, you want to ask something, feel free to, raise your hand, interrupt me. It's fine. It's supposed to be an enjoyable lecture. And so with that, I will start. So who can give me kind of brief definition, the best you can, of what is it to be a living organism in terms of genetics, not in terms of the whole philosophy of living. So anybody, what is it to be living in terms of genetics? Yes. You can reproduce, exactly. That's great, yeah, because reproduction is number 1 on our list. We need to survive as a species, or other species need to do the exact same thing. Anybody else? Okay, so we'll leave it at that. But essentially, you're all of these letters, ATCG. So this is not a specific sequence. I'm not trying to give you like a hack here. It's really just ATCG repeated. So your 4 nucleotides that code your human genome is repeated a lot of times here. Anybody can guess how many times this, how many letters are here? Wild guess. Hit me with a number. Size 5. Huh? 1 million? Oh, not so close. 30,523 letters. Now, what does that mean? What was the intention of this slide? Oh, can you see it? Okay, you can see at the bottom there. Okay, so it's just that to show you this percentage. If you had to have 1% of the human genome in slides like this, you need to have 9,843 slides to just have 1% of the entire human genome printed as such. This means that what you are, what humans are, what any living organism is an extremely vast sequence of alphabet, the human alphabet, the genetic alphabet, ATCGs, in repeat and specific sequences over billions of nucleotides, base pairs. So if you had to have 100% of the human genome, it would be 98,430 slides, exactly like that. So it's massive. And in 1990, they undertook the challenge, the Human Genome Project undertook the challenge to sequence the entire human genome. It took them 13 years and approximately $3 billion later, they had the full human genome sequenced. So that's interesting because that was the first time we really understood that one organism, which is us, we had all of its genetic information at our disposal. Now, the whole point of the next slides is to show you what we did with that information. So let's regress a little bit. I just wanted to know, show of hands, who here has a background in biology or just started biology? And so is it a majority or is there like other fields of study that are here guessing, like computer science? No? Okay, and synthetic biology? Okay, so I guess everybody understands at least the basics of biology. So the purpose of DNA is really to fulfill this central dogma, is DNA is the sequence, turns into an mRNA, which is this translational phase, and then it turns into a protein. And this protein now has a specific function that is dictated by that sequence. If the sequence is wrong, the protein will be wrong. That protein is essential for cellular function, for other processes, enzymatic, catalytic, whatever it is. That protein has to be perfect. Perfect comes from the sequence of the genetic sequence. There's other modifications that we could see, but that goes beyond the scope. And why is that important? Because it makes things like this. So if you can see here, there's 4 examples of what compounds are in existence because of a genetic sequence found in a natural source. And what do we do with these? So you have on the bottom here, paclitaxel. So Taxol, trademark, is from the Pacific yew tree. And so anybody has any idea what that drug is for? Paclitaxel? It's the first drug that was used to treat cancer. So breast cancer is currently being treated with paclitaxel. Came from a tree. Problem is that you needed about 3 full-grown trees to make enough compounds for one patient. So synthetic routes were developed so that we can actually develop enough of this compound to treat people. Next thing is opiates. So pain medication, they come from, I'm sure you already know, plants, and Papaver somniferum. So genetic sequences designed all the enzymes capable of eventually metabolizing, precursors into its final molecule of interest, which is an opiate. Then we have molecules like EPA and DHA, which are lipids. They're found in the fish. They're the reason why fish is really important, because we need these EPAs and DHAs in our diet. It helps form structures in our cellular level. And so, but that comes from the enzymes being able to catalytically convert precursors into final products. So this it's super interesting, but what does it mean in the global context? Well, it means that when we started off at $3 billion to sequence the human genome, the whole point was, well, every human is different. Let's make it more accessible. So they had to break down the cost of sequencing human genomes by a lot. And so I don't know if anybody knows what Moore's law is. I can't quote it exactly, but it's essentially, it's a law that we follow to see how well can a technology progress over time. And when we compare to how our cost per genome analysis is going, we actually destroyed that curve. We went so much cheaper than it should have been, and at a faster rate than we would have expected. Right now, you can sequence a whole human genome for about $1,000. So from $3 billion to about 1,000, that's a huge feat, which means now everybody can have their genome sequenced readily, and then we have all this great genetic information at our disposal. I'm gonna try to keep up here because I know I'm gonna go a little slower. So these are examples of companies that have built their entire core principle on genetics. So I'm sure you know 23andMe. Some of you might know CRISPR Therapeutics, Moderna vaccines, BioNTech vaccines as well, Ginkgo Bioworks, synthetic biology company, biggest biotech for synthetic biology in the world right now. And so these companies really core value genetics. So there's a lot to unpack there. Some companies even develop new therapeutics. So this is one of the viral particles that contain a gene editing, so RP65 coding DNA. So it's packaged in a viral particle so that it could deliver to eyes of people affected by the disease, and it could replace the mutated sequence and effectively giving them back their vision. So you can actually start curing certain diseases that have specific mutations in the coding sequence to give back the wild type sequence so that the protein can be expressed and therefore give back proper function. Other uses are genetically engineered models so that we can test in a lab setting a lot more efficiently, be a lot more proactive, and understand the effects of certain drugs a lot better. There's a bunch of other companies. These are not the only ones, so I was just trying to kind of give you an overview of who they are and what they do. But yeah, with new data comes new technologies. Anybody knows what this is? Correct? So what's the difference between the left and right? Just quickly. Exactly. So the one on the left is normal. The one on the right has sickle cell anemia. So anybody knows what sickle cell anemia is? It's a very simple, in terms of genetics, mutation. It's literally one letter that's out of place that causes the red blood cell to look from what you see on the left to making it look like on the right. And so that's a big problem because it doesn't carry oxygen, and people are affected by this all the time. So CRISPR Therapeutics actually initiated in 2019. Well, they got their first clinical data in 2019 showing promising results for treating this exact disease. And in 2023, well, they approved this therapy. So now individuals with sickle cell anemia can effectively get a genetically engineered therapy to help them replace that one bad nucleotide, effectively giving them back healthy red blood cells. So at the core of all of these is synthetic biology. And what is synthetic biology? These are definitions. I underline certain keywords that seem to come up in terms of concept, but really synthetic biology is an amalgamation of various fields and to create tools that are effective at doing genetic work. So it's the fine tweezers that we use to correct DNA or to modify DNA. That's really what synthetic biology is. It's a big toolkit. And so you have these definitions. I'm not going to go through them. But it gets more complicated because then you have the advancement of AI, bioengineering, tissue, in silico work, so tools and platforms, protocols. So all these kind of now blend into this kind of massive web of knowledge, and you have to start organizing that information a lot more effectively. So here's some definitions. You can read it really quick. It's just because I'm going to be using these keywords later on, so just in case we don't lose each other. Any questions up to now? Comments? Nope. So how do you do synthetic biology? What is the first thing that you have to think of? Well, you have to know how to visualize all this. You have to put this in a database. So these are some tools that we use on a regular basis that are specifically designed to showcase your entire genetic sequence so you can visually see what you're doing, understand why you're mutating certain things, how you're modifying it. And these tools are a great starting point for what we call the in silico work, understanding the in silico aspects of entire synthetic biology. Then you move into tools that are a little bit more developed that help you kind of search through multiple databases such as BLAST, UniProt, Twist Bioscience, and AdGene. A great company that we work with a lot is Twist Bioscience. They help you literally print DNA. So you send them a sequence of DNA, ship it to them, they'll ship you back a vial containing exactly that sequence in a tube. And so you can use that whenever you want. So it's really amazing what we've been able to do with just understanding at a nucleotide level. Every single field of production, of understanding, of medical sector has all expanded because of that fundamental knowledge that we've acquired on nucleotides. So this is why what you're doing right now, sitting in this class and learning about genetics, is literally the first step into getting all this information in your head and then seeing what you can do with it later on. And so, well, after that, so you have this virtual, on your computer sequence. You have the tools that you can kind of measure how good this enzyme might be or compare to other sequences. What do? Well, you choose your favorite plasmid. You guys know what this is. I'm pretty sure I saw it last class. So it's pretty much a vector that you can use to insert your sequence, what we call gene of interest, into something called the multiple cloning site. And that's right here. And so you insert your genetic sequence right in there, and then you can use this plasmid as a vector to transfect mammalian cells. Well, not puck, but like other vectors you can use. There's a lot of vectors. There's a large number of vectors. And so you can use these to pretty much effectively put your gene of interest into a model that you're interested in and study that exact same protein. And then you see how effective it is or not effective it is. There's multiple methods that you can use. So one is restriction digest, which is what you'd use for a PUC19 vector. There's BioBricks, sewing, Gibson assembly, keeps going. The list is long. There's a lot more tools that are being added every single year on a yearly basis almost to showcase how strong synthetic biology is and what novelties we can actually accomplish with it. Okay, then what? So you have this nice plasmid, you have your vector, boom. Well, you put this DNA inside the genome. So you're gonna put it either in a mammalian genome or you're gonna use it into a prokaryotic genome. So where you have this pretty much a plasmid, your design, floating around, and then the cell has no choice to keep it. So it starts expressing the protein that you want, and then you start studying it to do the experiments that Dr. Medcour is gonna show you on your exam. So, and it's all of this, you do all this work and all of this kind of information gathering just so you can switch one letter. So that's the letter that you see here. It's an A and T, and literally it turned into a T and A, which is what causes sickle cell. So this single letter shift causes the amino acid to go from a glutamine to a valine. And that single shift causes from this to that. And so it's a massive, massive, it's a small little mistake that causes a massive problem for humans. But this is not the case for all diseases. Other diseases are more complex, have larger sets of mutations and insertions, deletions, indels. So things like cancer, it's not usually one mutation the same way this is. It's thousands and thousands of mutations throughout your entire genome, which gives the cell the ability to become cancerous. And so there's other diseases as well that have genetic components. But eventually the goal is to use synthetic biology to start correcting these, to start removing a defective sequence and replacing it with one that is more normal or wild type, as we call it. So anybody know any cells that we currently use on a day-to-day basis? Single cells, it could be microbes, it could be any cell that we use commercially right now. Yes. Okay, E. coli, great. Yeah. Okay. Insulin. Okay. Yeah. So give me an organism that we currently use in everyday life. Like you guys consume this on a regular basis. Yes, yeast. Great. So okay. So E. coli, yeast, and then lactobacillus. So anything to make dairy products. So these are we've been selecting strains for a very long time. It's not any strains that we use to make these, right? We use specific strains. And so we've done some form of genetics there. We've done selection process. Now the next step beyond that is to turn these single cells, so such as bacteria, into biofactories. So literally hijacking their genome, making them produce things that we want to use commercially and we value a lot in a commercial setting. So bacteria can make proteins, antibiotics, drugs, yeast, medicines, enzymes, biofuels, cyanobacteria, enzymes, biofuels, and industrial chemicals, transgenic plants for therapeutic compounds, resistance as well for crops, and then microalgae, what we're doing for fatty acid, antioxidants, and animal feed. So nowadays, it's no longer enough to just take the bacteria that we currently, the organisms that we currently have, but we have to take them and then engineer new pathways in them so we can start meeting demand for this growing market. And so that's pretty much what this slide is showing you, is like the estimated value of biomanufacturing, so single cells becoming biofactories, in 2020 was about $18.28 billion. And so that's a lot. That's a lot of money. It's a massive market, and that's in 2020. So with more advancements in synthetic biology, it's only going to grow that much more. Companies like Codexis and Novozymes are great. By the way, I don't have any investments in any of these companies, so I'm not promoting them. I'm just disclosing what they do. That's pretty much it. And so why do we need to? As I mentioned, is because there's a lot of human-made problems that are reaching ecological maximas, so we can't actually use nature to actually solve these problems. So we have to start actually engineering the solutions to the problems that we created. And so that's going to be like a lock and key model. So problem X has problem Y, and that problem Y, if it's a biological solution, we'll most likely use synthetic biology to solve it. Yeah. So So yes, going back to my curve here that we destroyed because it no longer follows Moore's law, is really to decrease the costs of doing this kind of genetic work, increase the accessibility of high quality data. So data is one thing, but vetted data and then parsed data is very different. And then designing new ways of assessing this, creating specialized organisms is really the end goal for the entire process of research. But eventually you want to take this and implement it into a solution that's going to affect a global population. This brings me nicely into GC Lipid Tech. So it's the company that I founded with my brother. He's the CEO of the company and I'm the chief scientific officer. And so we've been working on this technology for a very long time, about 7 to 8 years now. And we started our company in 2021. Our goal really is to develop, well, actually, I'll give you the pitch, so make it easier that way. So welcome to GCLT, and let us tell you how we're going to engineer the future. Our mission is to grow the world's nutrients one cell at a time. And it's a very small, but we believe big difference, because every single nutrient that we currently have is not readily available forever. It's from a natural source. But we need to start creating alternative sources that allows us to create some predictability into the system. So this is why our mission is very clear. What we do, well, we're a synthetic biology company, fundamentally. That's really what we are. We're not any of the, we're not a feed company. We are a synthetic biology company because we have a platform that we use to apply our knowledge to engineer microbiology so that it can produce valuable metabolites. And this increases the predictability of that market, and that's a real goal. And hopefully I'll convince you throughout this that there is a lot of value to just doing simply engineering of microorganisms so that you can create more predictability in the market. Right now, pharmaceuticals, cosmetics, feed sector, they all rely on natural sources. They all rely on either animal protein, plant oils, plant proteins, plant pigments. And so as you imagine, all of those are from either the ground or the ocean, but it's all natural sources. So there's no really alternative aspect to this. There's like all proteins that are growing pretty big in the market these days. So that's a new way of shaping it. It's microorganisms that produce a large amount of protein, so we don't have to get it from a natural source. But there's a lot of other nutrients that are left pretty much forgotten in the mix. And the reason why this is really, really important is because we're at a critical deadline. Literally, I don't have much time, but yeah, so 90% of fish stocks are heavily exploited, and some of them are actually completely depleted. Like fish have gone extinct or at the brink of it. And so the World Counts is a great kind of a little timer for you guys go on the website. There's different kind of crises that we're facing, and they all put a timer that's been vetted by data and then supported by evidence. And so we have about 23 years, let's say 24 years before the end of seafood. And so if you imagine the oceans without seafood, well, you're actually imagining an entire ecosystem collapsing on itself. And if there's no ocean, there's definitely nothing else. So the whole ocean it's a big issue that we are actually facing today, which is acidification, overfishing, microplastics in the environment. So we have to start addressing the ocean as like this large commodity that if we don't take care of today will not be available tomorrow, and we're going to lose a lot of value in that. And we're not going to secure future supply chains for food security for the future generations. And so our target market, let me just take a sip of water here. Our target market for applications of microbiology is aquafeed. And the reason why aquafeed, again, is important is because it feeds 3.3 billion people on Earth. We're a lot of people that depend on fish as a source of protein. And if we don't have a secure supply of protein through seafood, well, it's going to be a big problem for us in the near future. There's even some studies that show that by 2037, so in really like 13 years, we're going to reach a permanent depletion. So we're going to start pushing the absolute boundaries of these ecosystems to the point where if they collapse, everything else falls like a domino effect. Right now, there's no renewable sources of oil, which is like fish oil. And I don't know if any of you take fish oil as capsules, but that all comes from a fish. Well, if you don't have fish, you don't have the oils. And so you need to find an alternative for that very valuable oil. And the whole goal of all of this is to put the fish back into the ocean and then use alternative sources as a way of supplying our entire manufacturing process. We just want to stop the timer. We want to make it ecologically friendly, predictable, and renewable. The market is actually quite big and is expected to grow to a tremendous size by 2030. So the number that you see here by 2030 is for the total addressable market. So that's the TAM abbreviation. It's about $320 billion market. and it's very big in Asia. It's also growing pretty prominently in North America. But these markets can't grow if they don't have constant supply. So you can imagine it's just like a paradox, chicken egg. It's like if we want to have a bigger chicken, but we can't feed it properly, well, how are we supposed to supply this entire market? So we're always trying to make sure that alternative sources are at the front line to take away the pressure that we put on ecological systems. Market validation. So recently, a researcher in Nova Scotia confirmed that microalgae is a sustainable source of fish oil substitute. So she did the study where she compared fish oil versus fish oil with microalgal oil, and showed that the fish grew the same rate, had the same body weight. And so that's a very important study because it shows that these alternative ingredients are not worse. If anything, they're the same, if not better, because of their carbon footprint, because of all the added benefits. And then recently also the government of Canada approved GMO and algae for a specific oleic acid oil. So these are great kind of little, success stories that help build the market. So as we become more friendly towards these kind of alternative sources, we become a lot more efficient, a lot more faster in development, and a lot more predictable. So the team is actually pretty big. We have 2 employees. My brother and I are also on the team, and then advisory council and strategic advisors that you can see here. We'll go through them all. We have a lot of partners. So building a company means that you're partnering with a lot of people, you're writing a lot of grants, you're getting a lot of support. And so you can't do it on your own, it's impossible. But it's really, really interesting once you start meeting people that spend their entire lives or their whole career trying to figure out one thing. If not figuring out one thing, it's literally helping the next best company into their pipeline so that you can actually succeed. So there's a really big network there. So entrepreneurship started off for me as like a goal and became like a passion. And now I'm just really happy to be a scientist entrepreneur because I get to see the best out of both worlds and through the network as well. So this is what I'm trying to like ask you. This is gonna, I guess, is gonna be part of the assignment, the few slides after. But just a little show of hands, how many of you have thought about the next step in your journey as a career? Okay, that's great. So you're thinking about it. Something's going on in there. You're not just passively doing this. Maybe, I don't know. Doesn't matter. What I'm trying to get at is that there's so many different things that what you're thinking about right now might not be what you're gonna do later on, or might not be the same thing that you want to do tomorrow. You might change fields entirely, but it's good to kind of have it in the back of your mind and just chew on it. How many of you would like to build a career in the field that you're currently in? Okay, that's good. Nice. And any of you thought of becoming an entrepreneur? Okay, so starting your own business and then just running it from the start. Cool. It's good to see because I would say back then, but not really back then, like let's say 10 years ago, becoming your own entrepreneur and running your own business wasn't really a thing. It was the classic route of go to school, get your degree, either become a professor, postdoc, or work for a big company, and that was really it. That was the trajectory. It's only recently that entrepreneurship has become a very central focus to a lot of universities. Yeah. Sorry? Don't worry, I'm going to convince you. I've been trying to start a business with this guy for a very long time. We've actually started businesses, but a side story. But the point is that it's an option. That's what I'm trying to get at. It's an option for you, so it's no longer just this, maybe if ever I had a lot of money, I'd start it. There's a lot of support. There's massive networks. There's accelerators, incubators, and I mean like worldwide. So anywhere you are, you can really be like, one day I want to become an entrepreneur. There you go. You can start it. So, quick little deep dive into my journey. So this is typically what you would expect, from bachelor's to master's to PhD and then postdoc, but that was not the case. And this is what it kind of looked like. So the arrows are kind of going in different directions at any time. But I started off my bachelor's, ended up doing a quick project in iGEM. I liked it. It was cool. That's the first kind of intro I had to synthetic biology. Then I decided that, I wanted to do my master's, ended up fast tracking. So I don't really have an hour going towards master's because we went straight to our PhDs. And then from there, I decided I want to understand what entrepreneurship. So I did a accelerator. I got accepted through a grant, a bursary for life sciences. And so pretty much drug development. And I went into the program, learned what entrepreneurship was, learned how, how this entire market evolves in the real world. Then I pretty much kept doing my PhD. I decided after my PhD, I was like, I still need to learn more about the industry. Finished my PhD, went to Charles River, became a study director, worked there for a lot, great work, super interesting, and then realized I'm spending a lot of hours doing work for other pharmas. I'm like, I want to spend time doing that work for my company. So left that company, great job, started GCLT with a lower salary because why not? Nobody needs to get paid more, right? And then from there, I got into my postdoc, which is what's supporting me right now as an entrepreneur. So, and we have our lab right across this, so in the glass, room that's there. That's our lab, and then we have our office space there as well. So your journey is never going to be a straight linear path. It's going to kind of bounce off different aspects, and each of these aspects will help shape your way of seeing your journey. So I encourage you, try a bunch of stuff. Don't be just expecting that this is the only path that you can follow, because that's what's going to be presented sometimes, but it's not necessarily what's going to be the reality. So this is the assignment I think Eunice is going to post. What the purpose of this, I'm just going to go to the next one. It's if ever you've had an idea, if ever you've had a passion that you're like, I don't know how to start exploring this passion, or I don't know what the value of this idea is, and you want to kind of just put it on paper in a very simplistic way, this is what I would suggest you do. And so it's really simple, what you are at stage in your career, what is the field that you're in, what is it that you want to develop? So it doesn't need to be what you're doing right now, but this is filled in with all of my criteria and my answers. But I would suggest that you swap it, because this will be the first version of you figuring out how your entrepreneurship journey will look like. Because you're going to do some research, and hopefully at the end of this, after all the research and all the information gathering, you can maybe convince yourself that there's something valuable there. And this is the way to kind of fish it out. It's a simple paragraph, fill it in, and see if it speaks to you in any shape or way. Yeah. 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 So, yeah. It's essentially like an elevator pitch. So it's this 30-second, set of sentences you could tell anybody, and they'll be like, oh, wow, okay, so you understand the problem that you want to solve. You have a solution that you've imagined, that you have an idea of. You understand the market size. You understand who it could impact, who's going to pay for it. So it kind of puts everything into one nice little context, so that if ever you have to reread this, you're like, I know exactly all the information I need to know in a summary format right here. And that's just going to be the first step, I believe, in figuring out how far you want to go with this entrepreneurship journey. But like Eunice said, you can use it for other things. It's just a way of creating succinct ideas in your mind. And on that, I leave you with Zig Ziglar's quote. I'm not too familiar with him, but I like his quote. So success occurs when opportunity meets preparation. And so I just added this little part, but although you may not control your opportunity, you do control your preparation. So preparation is this haphazard way of doing things, but you have to do it to completion and with passion. And as soon as you've done that, opportunities will start showing up. So I encourage you to, if ever you want to become an entrepreneur, start figuring it out as of now, because there's never a better time to become one. Yeah. Thank you for your time.
Okay. Was it good? You liked it? Cool. So I'm gonna try to do that every semester. Next semester. You? What do you mean you? Oh wait. Well, actually, you know what? It's actually serious. There's people that pay for this kind of stuff, and they make a lot of money. Usually, when they go to conferences, it's all paid for. When they pay their own stuff. But what did I want to say? So the reason why there's this assignment here is for you to kind of show me what you want to do, if you have an idea. And then if you send that to me, you get obviously the bonus points. But on top of that, I get an idea of what you want to do, and then maybe I can direct you to any professor or whatever, okay? So do that, think about it properly, send it to me, and we'll see. Sounds good? Thank you, Dylan.
Thank you very much.
Take care.
It was a pleasure.